Welcome everyone, and thank you for joining us to this other version of the International Society of Bone Morphometry webinar series. At the ISBM, we're focusing on all the aspects of quantitative assessment of bone across the scales, and we also include leadership panels and early career opportunities. Recently, we started having our working groups where we also host different techniques as HRQ micro-CT, spatial transcriptomics, imaging analysis, and bone micro and ultrastructure. If you want more information, you can join the website and register. This initiative is part of our Early Career Investigator Committee that is led by Matthias Wall and Anika Agrawal. And I want to take the opportunity to invite you to our next seminar that is going to take place in 1 month on Thursday, the 28th of August, 9 a.m. Pacific time, noon Eastern time, which will be led by Dr. Carl Jepsen, which is also from University of Michigan. We want you also to save the date. We're going to have our biannual meeting next year. It's going to be in London from July 3rd to the 7th. So save the date, mark it in your calendar so you can join us. Also, if you want to follow news, you can follow us in our new LinkedIn page. That's how we have the name. And lastly, we want to thank our sponsor and corporate partner for 2025, AstraZeneca, for their support for this webinar series. With that, please join me in welcoming Dr. Annemarie Lang. She is a tenure-track assistant professor in the Department of Orthopaedic Surgery at the University of Michigan. Her research focuses on the role of erythrocyte development and function in musculoskeletal health, injury, and disease. During her postdoctoral training with Dr. Joel Boerckel at the University of Pennsylvania, she discovered that bone fractures stimulate localized erythropoiesis, which modulates oxygen levels and support their tissue repair. In this talk, Dr. Lang will share her insights from her scientific journey, from her PhD work on HIF signaling in Berlin to establishing an independent research program in the US. The presentation will highlight how bold scientific questions and interdisciplinary international collaboration have shaped her career and continue to drive her research forward. Welcome, Dr. Lang.
Thank you, Carlos, for this really nice introduction, and also for thank you for having me. And yeah, I'm really excited to be here today and give you the presentation. And I think, as I was already introduced, the stakes are high, I would say. So let's see what we can do in the next 30 minutes. So yeah, the title of my talk will be "Breathing Life into Bones: the Local Oxygen Microenvironment During Fracture Healing." And in order to give you a little bit an outline what you can expect from this presentation, so I will give you a really short intro into my personal journey, because to be honest, I always loved when listening to these kind of more career development focused talks, if people told their own stories, because I felt that was really inspiring and also, yeah, gives you maybe also some hope or inspiration. Let's hope inspiration. Then I will talk a little bit about the background, like hypoxia, HIF signaling, and how this all came together in fracture repair, what the idea is there. And then I want to come back to what I did during my postdoc and how we now redefine the local oxygen microenvironment. Okay, let's get started. So my personal journey. So actually, I come from a really small town in Germany. It's southeast. It's former GDR. Actually, it's Freiberg in the beautiful Saxony. And I studied veterinary medicine because my biggest dream was actually to heal the bones of horses. So at that time, I really liked horse riding, and I was super interested because fractures in horses are actually typically fatal. So there are no good ways to increase healing and to actually get it stabilized. That's why I decided to get into veterinary medicine. However, how life always plays, during studying, I was actually not so into the whole equine medicine. And I got really interested in immunology and started to join the lab of Professor Michael Schmidt in doing an internship during my studies, and came into contact with osteoarthritis and cartilage, actually, which led me to pursue a doctoral thesis on osteoarthritis gene therapy. And this kind of kicked off my whole scientific career, I would say. And here on the right side, I also have a picture from, some of you might know him, Farouk Rai, who was an assistant professor at the University of Washington and is now in the Emirates. And he actually did his PhD in Berlin in the same institute before me and also worked on OA. So I also found this bridge actually later. Yeah, so since I wanted to really go more in the direction of a scientific career, I thought like, okay, let's add a little bit more to it rather than only a doctoral thesis. Let's go for a PhD. So I joined the Berlin-Brandenburg School for Regenerative Therapies because, like, yeah, as you could guess, gene therapy has a lot to do with regeneration, and I wanted to explore this topic and the potential of translation of regenerative therapies a little bit more. And I also wanted to get another field of research, or like to explore another field of research, because those of you who know, working with cartilage can be really frustrating a lot of times. So I actually wanted to expand my horizon and go towards fracture healing. So I did a PhD on hypoxia signaling and bone fractures, and then later on stayed for 1-2 more years as a postdoc and wrote a habilitation. A habilitation is like a postdoctoral thesis. Yeah, and then I was at a point in my career where I thought like, okay, either I go up for professorship or I do a trip to the US. And actually, that's what I did during the pandemic. It's not the best thing to do, right? During a pandemic to move to the US, especially a big city as Philadelphia. However, it was the best decision in my life, I think. And so I pursued a postdoc and focused on oxygenation and bone fracture. So you will hear more about this later. And I also received a Marie Curie Global Postdoc Fellowship, which led me back to Dresden and to Germany. And then finally, I applied to the University of Michigan and now in January started my own lab in the Department of Orthopaedic Surgery. So you can see that I kind of have a linear pathway, but maybe not in all aspects. So, and I will come back to some of these steps in between. And for those of you who are interested in going ever to Europe to do science, please check out the Marie Curie fellowships. They are amazing. Okay, let's jump into fracture healing. So fracture healing is divided into different phases. We have the initial phase, we have the soft callus phase, hard callus, and remodeling. And the beauty of fracture healing compared to cartilage injuries is actually that bone injuries can heal really well without any scar formation. The big question is, why is that? So once the bone breaks, the vasculature is disrupted and the bone marrow cavity gets filled with blood, and this clots, which forms like an hematoma. And this hematoma was supposed to, or is supposed to be hypoxic because the vasculature is disrupted. And when I started my PhD, this was literally where I started off, was like this understanding that we need this low oxygen in order to kick off all the regeneration regenerative processes which come afterwards in order to ensure that the bone can fully regenerate. And this was actually based on the understanding that the initial phase is hypoxic, was based on really old papers which were done by Brighton et al., also from the University of Pennsylvania, in 1972, where they looked into oxygen tension of healing fractures in rabbits. And here you can see this table. So they looked at different days. They had an electrode, which they literally put into the fracture gap or like all around tissues. And so they measured at 4, 7, 10, 14 days, so a lot of different days, and then different kind of areas. But here's the hematoma, which is really interesting. And so it showed like 6.25 millimeter of mercury, which, as you will learn later, is really low. So this was actually the start. And what they did is, and I think it's really important to understand, they used this rabbit fibula. Let me just take no, wait. Just want to show you with the highlighter. Yeah, so they used the rabbit fibula and they actually made the cut here at this area, and this is where they measured the oxygen. Okay, so hypoxia is obvious then in this kind of scenario, and this typically leads to HIF signaling. So this was the next indicator which actually contributed to the whole understanding. HIF signaling, like jump in. HIF is hypoxia-inducible factors. So typically every cell in our body expresses HIF-1 alpha, and this is constitutively expressed. However, under normoxic conditions, the prolyl hydroxylase is active, which is an enzyme which marks HIF to be actually degraded by a proteasome. So it's constantly degraded in the cells. However, the PhD enzyme is dependent on oxygen or iron or oxoglutarate, meaning under hypoxic conditions, it's actually inhibited because it's missing the oxygen. And so HIF can be stabilized. It's translocating into the nucleus and forms complex with HIF-1 beta in order to actually guide different kind of genetic sequences, for example, angiogenesis. So it was shown in fracture repair that this is actually really important in order to allow for regeneration. So in 2007, the group around Tom Clements actually showed that if you knock out HIF in bone regeneration, you actually reduce vascularization and therefore reduce bone formation. So it's actually really important to have signaling, HIF signaling, which means like we again have this kind of correlation between there's hypoxia, there's HIF signaling, so it must be somehow helpful in order to have bone regeneration, especially in a context of revascularization. Because here on the right side, you can actually see this is a large critical size defect in a rat model, and all of this is filled with vessels, and these vessels are really essential in order to allow for regeneration. So which means kicking it off with HIF signaling is not that wrong. And during my PhD, what I actually did is we applied deferoxamine. Deferoxamine is an iron chelator which activates HIF signaling, and we applied it during bone regeneration and found that we could actually significantly increase vascularization and also bone formation. So underlying this importance of HIF signaling during fracture repair. In parallel to this, we started to develop an in vitro fracture gap model where we combined these tissue engineered bone equivalents, as you can see here, together with like artificial fracture hematomas in the culture dish and cultivated it under hypoxia or even normoxic condition, and found that especially under hypoxic condition, we had a stronger induction of regenerative cues. And this brings me back to this kind of correlation. We have HIF signaling in fracture repair, and we have this common understanding. However, when I wrote the introduction to my PhD thesis, I sometimes really struggled to find additional literature in addition to the Brighton papers from really early 70s, which showed that it's really hypoxic and how this interacts. So I was actually wondering, how can cells survive in this environment, and how can they actually take it in order to regenerate bone? So what are the really mechanistic cues in that? And then there was always like this I would say smaller question mark, like, is it really hypoxic? Do we really know? I don't know. Maybe we should test some other methods. And this actually led me then to pursue my postdoc at the University of Pennsylvania. Before coming to this, I want to give you a really quick intro into oxygen tensions, because I already started off talking a little bit about hypoxia, normoxia. However, how is it really in our body? So in the ambient air, we typically talk about normoxic conditions, which is around 21% of oxygen. If we come into the body, this is actually reduced because the cells are using it. So in arterial blood, we have 13.2% oxygen. The spleen is like an oxygen reservoir with 8 to 10%. And then you can see in all other organs that it's different, actually. We talk typically, or we define hypoxia as under 5% of oxygen. And when we look into the bone marrow, which is of interest for us, we can actually see that in humans, it has been shown that it's around 6% of oxygen, or in mice, 4%. And there was this nice study by Spencer et al. Who looked into a cranial window and really down into the bone marrow extravascular spaces and found that the oxygen tension is actually really low, around 10 millimeter mercury, something around this, which actually aligns with the 1 to 4%. So there's also a heterogeneity in the hypoxic niches in bone marrow. So in order to actually answer my question, I use a fracture model, which is a femoral fracture model in mice. It uses an extern fixator system. So we have an extern fixator. We can place it in the femur. We create a gap in the middle. We have different stiffnesses that we can combine. And it has a really nice healing trajectory. So at 3 days here, this is Movart's pentachrome staining. Yellow is the cortices. Bone marrow is in dark red here. And here in the middle, you can nicely see the fracture hematoma that we can also find in mice. At 7 days, we have the invasion with cells and stem cells. At 14 days, we already have mineralization in the gap, which is then more pronounced at 3 weeks. So it's a really nice model. And we are particularly interested in the early phase. So let's see. How can we actually stain hypoxic cells? So I established EF5 staining to mark hypoxic cells in the fracture. EF5 is a nitroimidazole, which is reduced by nitroreductase enzymes under hypoxic conditions and then forms intracellular adducts. So what we actually see, or what we do, is we have a cell with high oxygen. The nitroreductase enzymes reduce the oxygen which is available. However, under low oxygen conditions, some of these enzymes are actually available. So if we add a nitroimidazole like AF5, it can be reduced by this available nitroreductase enzymes, forms these really stable EF5 adducts, which can be then stained. So actually, the more EF5 adduct staining we get at the end, the lower the oxygen concentration around the cell. I applied this method, or I applied the compound in mice. And then after 2 hours, I dissected the bone marrow and the spleen and analyzed it via flow cytometry. So here's the spleen. You can see this is the cell count. And then on the x-axis, you have the EF5 staining intensity. So the spleen, as I told you, is an oxygen reservoir. So there are literally no cells positive for EF5 because it's not hypoxic. So you can nicely see that the control staining in the histogram is overlapping with the treated sample actually. In bone marrow, it's a different picture. In bone marrow, we can actually see a positive staining in EF5, and it's around 50%, 50-60% of these cells are positive for EF5. EF5 is most sensitive under 3 to 2% of oxygen. So it's more sensitive at really low concentration, which aligns nicely with the literature where it was shown that it's like 4 to 1%. So not all of these cells are in like really hypoxic niches. So I then applied this to the fractures and wanted to actually dissect out all the hypoxic cells in order to see and sequence those to find out how these cells actually handle these really low oxygen concentrations. So in the extern fixator, the beauty of the model is actually we can distinguish between a gap area, which is in between the middle pins, and an ipsilateral bone marrow area, and then compare it also to the contralateral bone marrow. So I applied it to all the controls, as you can see here. And in dark gray, you can see the no EF5 background control, then the ipsilateral and contralateral bone marrow, which is the same image as we had before, like 50-50 almost. And then for the fractured one, I was actually expecting to have it higher. However, what I found was that there was literally no EF5 positive cell in the gap. And this was really surprising. And first I thought there was like a technical mistake. So I redid it multiple times. And as you can see here, it was literally almost the same. And then I repeated it at 5 days and also at 7 days. You can see there is a slight increase in EF5 positive cells over time. However, it's still significantly reduced. So based on this data, we actually started to wonder, okay, maybe the fracture gap may not be as hypoxic as we always thought. So in order to make sure we really see what we see, we actually used a second method, which was a direct oxygen measurement method, where we use phosphorescence lifetime imaging or phosphorescence quenching of the oxy-4 compound. So the compound is injected 1 day prior to fracture, so that if we make the osteotomy, the compound can actually get into the fracture area, and then it's done as an endpoint measurement. And here you can see, so with the laser, you can actually work up and down on the bone and measure the tissue oxygenation. And using this and going into the gap, we actually found in the contralateral bone marrow, it was around 30 millimeter mercury, which is actually the same that I showed you a couple of slides before as determined in the literature described. However, when we went into the gap area, so when the bone was fractured, we found this elevated oxygen content. And then we did measurements over time. And also here we found that after 7 days, it was still elevated. And then it got back to the normal bone marrow environment at 14 days post-fracture. This actually let us wonder, okay, is there maybe a dependence on the vasculature? So is there something about the vasculature that we overlooked? So here you can see in the graph, it's the oxygen concentration again on the y-axis, and then the time. And this is just the oxygen graph that I just showed you, just summarized here. So we found there is decreasing oxygen tension. However, when we looked at the revascularization capacity, we found actually an inverse relationship. So we measured the endomycin area in all of the sections that we had, and that we also measured for the oxygen tension. And we found that the percentage of endomycin positive area increased over time. So it's actually independent of the vasculature. So quick first summary in between. Our first part actually shows there's differences in the oxygen tension as we originally expected. And the big question is actually, what is the underlying mechanism? Because we also found that it's independent of the revascularization. And one of the ideas was that during the fracture process, it's actually bleeding into the area and into the bone marrow, which means there is a lot of oxygen coming in just by having red blood cells available or locally there. However, these erythrocytes don't survive for that long as like 5 or 7 days, for example. They typically survive, I don't know, 1-2 days maybe. So there needs to be something which actually locally stores the oxygen or keeps the oxygen locally. So this is where we got really excited and explored a little bit more towards the direction of erythropoiesis, so erythrocyte development in the bone marrow. Here you can see the trajectory, how red blood cells develop in the bone marrow. We typically have in hematopoietic stem cells, which becomes a more prone towards the erythroid lineage precursor cell. It's a megakaryocytic erythroid progenitor cell, which then becomes a pro-erythroblast. An erythroblast, it throws out the nuclei, jumps into the circulation, becomes a reticulocyte, which is like the young erythrocyte, and then an erythrocyte. And during this development phase, these cells express different kind of surface markers. So for example, CD71, which is the transferrin receptor 1, which is an iron importer molecule, which makes sense because these cells need a lot of iron. And then there's another marker like TER119, which is stained in mice, or CD235A in humans, which is just something about the membrane composition. And then CD45, which is like reduced over time during the development and is more stem cell marker. And here you can nicely see in the bone marrow like a live erythropoiesis happening literally. So we have these precursor cells which have different degrees of CD71 or TER190 staining. And then when it comes towards the vasculature, actually you see that all of the TER190 positive erythrocytes or reticulocytes don't contain any nuclei anymore, and they are typically thrown out here on the side. So we used this staining and applied it actually to our fracture area. Here you can see the pins. This is the femur section, and in the middle you can find the gap. And in white we have the CD71 stained, and in magenta we have the TER190. And we can nicely see that here in the area of the gap we actually have a higher abundance of these cells and an accumulation of erythroid precursor cells close to the injury site. So we quantified that on different kind of levels. For example, we also used single cell RNA sequencing, and we found that especially the pro-erythroblast population is enriched in this area. Here, this was from the single cell RNA sequencing, but also in flow cytometry, we found the same vector. And that's actually interesting because those cells, when we looked back into our data on the ER5, what we found is when we gated first onto the CD71 positive and TER190 positive population and graphed it onto the graph with the ER5, we can see here in magenta that especially these are the cells which are negative for ER5, and while the CD71 negative and TER191 negative cells are actually those which are positive for ER5. So this was in a this is a picture from a normal bone marrow sample, actually. So we concluded from this that these are the cells which literally contain the oxygen. And this makes sense on so many levels. So it's sometimes really fun when you find something which makes sense, but it's still really surprising, actually. So these cells produce a lot of hemoglobin, and hemoglobin consists out of iron, which is integrated into the heme structure, and then it's combined with 4 globins, and it's actually able to bind oxygen, and it's also really pulling in oxygen. And iron is transported into the cell by the CD71. So these cells literally have the whole machinery in order to bind oxygen. But the big question was, are they really the ones, or are these the cells which might concentrate the oxygen locally in fracture repair? So what we did is we injected a CD71 antibody right after creation of the fracture gap in order to actually reduce the iron import capabilities of these cells and therefore then reduce hemoglobin synthesis. And we did, again, the measurement of the direct oxygen like 3 days post-fracture. Here in this image, you can see the isotype control. So we had the contralateral bone marrow, we had the fractured bone, the gap area, and then in the CD71 treated, we found that in the contralateral bone marrow, it was comparable to the control. However, in the gap area, we found a significant reduction in the oxygen tension, which was really incredible because we inhibited a cellular process by just importing iron, but therefore reduced the whole oxygen content in a tissue. And we also looked what would that do to the environment actually, or to the healing environment, if we really turn it into hypoxia. And so we looked 14 days post-fracture after injecting the antibody only once during the creation. So we didn't re-inject the antibody. And we found that in the CD71 treated animals, we had an increase in the bone volume fraction and also in the general bone volume formation. And a really striking finding was also that when we looked in immune fluorescence staining, so here in green, you can see the N-myosin staining for the vasculature, and in white, it stains the osteoblast or osteoprogenitus for Osterix. And typically what you see 2 weeks in this model is like, it can be healed really nicely. It can be more like this example that you already have vascularization, you already have some mineralization. However, in the antibody treated animals, what we found is actually a really increase in the revascularization, and all of these gaps were already mineralized with only small cartilage here on the side. And this was also a really intriguing finding, and also suggested that what we originally thought that hypoxia is important for fracture healing is not wrong. It really helps the regeneration. However, in in the natural process is actually a little bit different. So to summarize the last findings, so as I said, we have to rethink the oxygen model for fracture repair. We created or developed this really unique oxygen monitoring pipeline that we can apply in small animal models too. And we found that upon fracture, we have an increase of CD71 TER190 positive erythroid precursor cells, which can scavenge the oxygen locally. And if we inhibit their capabilities to bind oxygen, we recreate hypoxia and can actually increase regeneration. And this is now the foundation for my current research program here at the University of Michigan. And I'm really excited to dissect the whole process and learn more about these cells, and especially how erythropoiesis intersect with like musculoskeletal disorders. So some final thoughts, because this is also a little bit about career development, is so based on my journey, what I can say is like embrace challenges and be resilient. So typically I got frustrated a lot of times during my whole journey, but it's like I typically give myself a couple of minutes or like even a day, and then get back up again and just try it again and just, be resilient in the whole process. And then innovation begins with the courage to doubt and to probe the obvious. So I think in the beginning, when I started to discuss with people about the hypoxic environment, that's really something that we had in our minds at the time. And now this might be a little bit changed or like redefined actually. And to be honest, when you look back at the initial slides that I showed about the Brighton study, what they used was the rabbit fibula, and the rabbit fibula does not really contain bone marrow. However, what we looked at was actually the femur, which contains a lot of bone marrow. So I actually think that there's a difference in which bone is broken. And that's also something I want to explore in the future, if there is a difference in the erythropoietic activity and therefore in the oxygen environment during fracture repair, which then can also influence your fracture healing outcome. And then, one of the most important advices that I can give is really find your mentor and your champion. You need someone who supports you throughout the whole path, and who also helps you to get into the next steps of your career, and who always supports like the ideas that you have. And they might not be that crazy, or it might not be or it might be more incremental, but it needs to fit actually your needs. And when I went for my postdoc, it was, yes, I had all these questions, but I also decided that I think Joel is the perfect mentor for me, who can guide me through the whole further career. And I think that's a decision everyone can make for their own. And with this, I like to actually thank all the collaboration partners and also the Boerckel Lab. And I'm really excited to start now my journey, my own lab. And we already started to make like almost as nicely funny pictures as we did at Penn. And with this, I'd like to thank you for your attention. I'm looking forward to the question. And please provide feedback. I'm always happy to take feedback from my presentations and improve them. And thank you.
Thank you so much, Dr. Lang, for the amazing presentation. It's great to see the whole pathway of your career. Is there any attendee that want to ask, have any questions? Dr. Akins, go ahead.
Hi, Annemarie. That was a great talk. Nice to see you. When you did the local injection of the CD71 antibody, did you see any other type of side effects on the mice, or did you also look at the bone quality of the newly formed bone?
So we did not see any other side effects, and I know CD71 plays a role in other cells too. That's true. The antibody has a half-life, or what I found in the literature, it's typically present like 3 to 4 days in the body until it's completely gone. So we injected it right at the creation of the gap. So we created the gap, I closed up the wound, and then I injected the antibody locally, which means the antibody is present like 3 days post-fracture, so the time point that we are interested in. And at this time, the cells which are present in the fracture gap are mainly immune cells, neutrophils, macrophages, and the erythroid cells. And looking back into the sequencing data, especially the erythroid cells are the highest expressing cells of CD71. So the likelihood that we more specifically target these cells is really high. I don't think that we have any effect on the osteoblast just by one injection, to be honest, and this early time point. But we are now starting to conduct repeated injections, and I think then it will also affect the osteoblast, for example.
Yeah, thank you. I'm looking forward to that.
Do we have any other questions or comments for Dr. Lang?
How did you go about kind of like reframing your research question when you came up against what seemed like kind of like the opposite effects of what you were anticipating? And like, how do you recommend kind of creating like a thought process for when you come up against kind of counterintuitive results in your research? Because I know you said at first you're like, well, I must have done it wrong. And then you went down a path where you were really intentionally investigating these alternatives. So how did you kind of create a plan for yourself and like follow that workflow for thinking that creatively? Is that even like does that make any sense as a question?
No, absolutely. I mean, it was in the beginning. This is why I also mentioned in the beginning, I had this Marie Curie fellowship. So first of all, it's an amazing fellowship, but also I wrote the grant for it. And the first sentence was fracture healing is hypoxic and we need hypoxia. And so the first experiments I did for that was literally showing the absolute opposite. And I was like, yeah, well, I mean, okay, hypothesis done, literally. But I think what we did is, first of all, we went a little bit further down and like tried to really come up with different techniques to validate our finding, because I think that's really important, just based on the EF5, and then I think that's really hard to say. So there needs to be some parallel method that it can be applied, and we found it on so many different levels that it was actually repeated. And then we started to wonder, okay, what's this? So I literally didn't do anything further what I wrote in the grant, and I just took the other direction because I became really excited about. And I think that's the beauty of if you have a postdoc environment where you can be more creative. I'm not sure that this would have been a question that I could have done during my PhD, just because, I mean, doing a PhD, you are much earlier in your career. You can start some side projects maybe, but you cannot start a whole new research program, actually. You need some more independence. And now, going now the next step into my own lab, I can now fully embrace it and like really look into erythrocyte development and function in musculoskeletal. So I think it's also about patience. Sometimes you have these questions which come up really early in your career, and then they will stay in your mind. And at a certain time, you have the right space, an environment where you can just test it. So that's and be positive. So I think one of the biggest or like one of the strengths of Joel was always when we had these contradictory findings to make it in something so positive that you just could be excited about. And that's also something I now try to translate to my group, that a negative finding is also something really good. And so, yeah.
Thank you.
Yeah.
And very nice start. Yeah, very nice presentation. So I have a question, after injecting the CD71, antibodies, you trapped basically the chelated the oxygen. So, and by the ferrous, the iron molecule. So iron is also a cofactor of PhDs, protein hydroxylases. So have you ever seen or have you ever, got anything changes in PhD level like PhD 1, 2, or 3? Have you checked that?
So I haven't checked that, but that's actually an open question because the PhD would then come back to HIF. And I think so. My idea or hypothesis is actually HIF is still is present in fracture and is super important. I'm not saying that anyone was wrong before. It's more like I think it could be not it couldn't it could be more the iron deprivation in all other cells because the erythroid cells are literally stealing it from the other cells that you stabilize HIF rather than maybe the oxygen tension that is a striving factor in this. So yeah, I think the
I was just wondering.
CD71 could
I was
also
just wondering
increase
that
HIF.
it may be just, a novel function of PhDs rather than HIF and HIF,
Yeah.
isoforms, but yeah, this is very wide,
Yeah, no, absolutely. And that's also something I'm wondering about the deferoxamine, for example. Typically it's always HIF stabilization and or supposed to be HIF stabilization in the whole environment, but maybe it's really also due to the erythroid cells and reducing the hemoglobin and also in reintroducing an hypoxic environment. So also on an oxygen level than only on a cellular level.
Yep. Thank you.
Yeah.
Do we have any other comments or questions? All right. Thank you so much, Dr. Lang, and thank you everyone for joining us today. And please join us in our next month webinar.